All right. Thank you all for coming today and get started. Very lucky today. We have Professor Sabeeha Merchants joining us from UC Berkeley. She's an internationally recognized leader in algal biotechnology, algal genomics, lots of amazing work that we'll fortunate enough to see today. She's led lots of sequencing efforts over the years. A lot of Chlamydomonas fans in the audience, I hope. Chlamydomonas reinhardtii, this amazing green algae that's been a workhorse for so much important discoveries over the years. Sabeeha led the sequencing effort for this, the original sequencing effort that's been this amazing resource to the entire community over the years. She's also done a lot of work adopting other non-model algae and sort of turning them into model systems as well. And so lots of people are starting to work on all these other amazing systems because of her work in these fields. For all this research, she's received many awards over the years, and I'm not going to have time to go over all of them, but I'll just mention she's a member of the National Academy of Sciences and the American Academy of Arts and Sciences. She received a Humboldt Award from the Alexander von Humboldt Society, which is pretty cool. And she also, in addition to all of this, she also does lots of really important service for the community as well. So she's editor-in-chief of the Annual Review of Plant Biology, and she was editor-in-chief of Plant Cell as well. And she has a very busy schedule, so we're very lucky to be able to get her out here. As part of this trip, this is actually the third talk she's given on this trip. She gave 2 other talks in Indiana before this, so I'm assuming you left the best for last, right? So please join me in
Thanks so much for the invitation. It's been fun so far, and a special treat was, getting a chance to talk to a colleague from grad school days. So that was nice to have the chance to see Jim. I made up this title, because that gives me the excuse to talk about like 3 different projects in my lab, and so we'll see how it goes. Okay, so. The word algae is actually a common noun. It doesn't have any, phylogenetic meaning. And what I've shown here in this tree of life, all the colored branches represent algae. And you can see that, in fact, they're found in many lineages. And my group work is focused on the green algae. So this particular radiation is a product of the primary endosymbiotic event, where some progenitor eukaryotic cell engulfed a cyanobacterium that then got established as a plastid or a chloroplast. And that happened a billion years ago or more, and gave rise to the radiation shown here, which includes the land plants and then also the green algae with which I work. And I mentioned this because the relationship between, this radiation and the lua is, probably as similar as the one between the green radiation and, say, animals, right? Except for the fact that these other radiations result from secondary and tertiary endosymbiosis. So there is the plastid that might be in common. So anyway, the reason that we focus our attention on the green radiation is because the work that we do in algae can benefit from discoveries that are made in the land plant systems, because there is a common ancestor. And then vice versa, we hope that the discoveries that we make have an impact, for example, on land plants and on agriculture and crop technology as well. So as Frank mentioned, I started my career working on the so-called model organism, Chlamydomonas. But today, because of the advent of high throughput sequencing and the advances in sequencing technology, plus the ability to modify any genome of interest, really you can make any organism a model organism because you have access to so many tools today. So these are some of the other organisms that we have worked on. The top 3 are in the chlorophytes. So Chlamydomonas and Dunaliella, you could say, are sort of more like cousins, except that they're still 500 million years apart. And then more recently, we started working with these, with Oxynochlorella, trabuxiophyte alga, and that's more like 850 million years apart from Chlamydomonas. So really divergent, but again, in the same lineage. So I'm going to try to tell you 3 stories today. The first one results from the work that we've been doing actually for decades, trying to understand how algae and then by extension land plants as well acclimate to deficiencies in trace nutrients in the environment, because these and by that I mean metal nutrients in my lab, so copper, iron, zinc, manganese, etc. And these are all important for metabolism, and many environments are deficient in these micronutrients, so there's biochemical acclimations to this, and we wanted to discover that. So in the context of the photosynthetic apparatus and the thylakoid membrane. So I'll first talk about that. And then secondly, I'll also talk to you about how our work has resulted in fundamental discovery, which I hope will one day enter the textbooks. And then finally, if we have time, we can talk about how we've been using algae for biotechnological application. All right. So first, let me talk about Dunaliella species and the mechanisms for handling iron deficiency. So I'll just remind you, this is sort of the, this happens to be Chlamydomonas, I think that's up here on this graph. This is the elemental composition of, Chlamydomonas. But really, this would look the same if I showed you any other alga or almost any other organism. We don't see hydrogen and oxygen here because, maybe they'd be, back up there on the, on the wall, because of course, most of our biomass is really water. But importantly, you'll see elements like these, iron, copper, zinc, manganese, etc., that are present at very low levels. But this is actually what made life possible, because these guys are the ones that are important for catalyzing all the biochemistry that is necessary to allow life to happen, especially bioenergetics, because ultimately that's what life is about, the redox reactions. And you can see here that if we drop out one of these nutrients from the medium, these cells get growth limited. Okay, so iron was one of the earliest elements to get incorporated into biology, because at the origin of life, it was very abundant. So it was used for a bunch of chemistry. And you can see, in fact, that it's very versatile in terms of redox potential and the types of molecules that it associates with, for example, with organic cofactors or inorganic cofactors. And it can span a huge redox potential. Okay, so as I mentioned, it was incorporated early on in the origin of life, but then after the great oxidation event, when oxygen became more abundant on the planet, the ferrous iron, which is very soluble and bioavailable, became ferric, and of course, less soluble and less bioavailable. So today we're faced with an environment that is iron limited for essentially, actually nearly all forms of life. And this picture here shows you the iron prevalence in agricultural soils. So this heat map, the blue shows you low iron conditions. And you can see that, a lot of Western Europe, for example, can be iron deficient. So 30% of agricultural soils are iron deficient. The same is true of the oceans. These are, of course, estimates based on dust deposition from landmass. And you can see again that the equatorial Pacific, the Southern Ocean, and of course, seasonally other oceans as well can be iron deficient. So half of global primary productivity actually occurs in the oceans, and then 30% of that can be iron limited. All right, so my group a few years ago decided to look at an organism called Dunaliella species. And the reason we were interested in this is that compared to Chlamydomonas, Dunaliella had a much lower requirement for iron and could still grow under iron deficient conditions. And this is actually an organism that is abundant. It's what we call cosmopolitan. It's found all over the world. These orange and green dots show you places where people have isolated Dunaliella. It's actually an extremophile. It's halotolerant. And the stars here show you the two species that we worked with in the lab, whose genomes we sequenced. The green one is Dunaliella tertialecta, which came from Oslo Fjord in Norway, living in about 0.5 moles. And then the orange one, the Sinai Peninsula, and that's 2 molar sodium chloride. And it's actually used a lot in Israel for biotechnology as a source of carotenoids and glycerol. And at the time, Vital Davidi, who had in fact, worked in Israel on Dunaliella, joined my lab as a postdoc, and was interested in exploring its mechanisms of surviving iron deficiency. So of course, the first thing we did was sequence the genomes of the two organisms. They're actually much larger than Chlamydomonas, about 300 megabases, much larger genes, much larger introns, and so on, but essentially very similar coding capacity. And when we then we did transcriptomics and proteomics. I'll just show you like a couple of figures from those experiments. On the left-hand side are results from the transcriptome, and then on the right-hand side, it's recapitulated in the proteome. So this is your typical MA plot. Anything on the right-hand side means the gene is very highly expressed. And then if you look at the y-axis, that's the log2 fold change. So anything that's, the very top or very bottom here, then are very dramatically regulated. So I'll just draw your attention to two proteins here, flavodoxin and something called TD. So flavodoxin is a redox protein. It accepts, it's been known in bacteria for a long time that it can replace ferredoxin, which is an iron sulfur protein, very abundant in green organs of a plant or in green algae because it accepts electrons from photosystem 1. So Dunaliella is a strict photoautotroph. It grows exclusively by photosynthesis. So it needs its photosynthetic apparatus to grow. And this flavodoxin comes up because it can uses flavin as a cofactor, an organic cofactor, and it can replace the ferredoxin in electron transfer at photosystem 1. Okay, so I'm not going to talk to you about that anymore. These, by the way, these are classic markers of iron deficiency, essentially, again, in all forms of life. This is a ferric reductase that solubilizes ferric iron and by reducing it to ferrous. And then IRT1 is a transporter. The other protein of interest is this one here, thylakoid iron deficiency inducible. And this is what I'll spend the rest of this part of the talk on. Okay, so when we look at what things are downregulated, again, you'll see this is the gene for ferredoxin. It's retrenched because it's the most abundant iron-containing protein in the cell. It's a 2-iron, 2-sulfur protein. Ferritin, of course, an iron storage protein, is downregulated. And then these guys here, PSA and LHCA, these are the subunits of photosystem 1. So in the photosynthetic apparatus, we have two photosystems, photosystem 2 and photosystem 1. Photosystem 1 houses half the iron in the photosynthetic apparatus, right? So it's a big sink for iron. So the cell responds by retrenching photosystem 1. And you can see that with respect to gene expression, PSA stands for the genes for the photosystem 1 reaction center. And then LHCA, these are the light harvesting chlorophyll binding proteins that associate with photosystem 1 to harvest light or bring photons into the reaction center. So you can see that all of these guys are downregulated. And again, it's recapitulated at the protein level. Here are the PSA subunits, for example, and then the LHCA subunits. And then you can see increased expression at the protein level of TD and flavodoxin. Now, when we look at, so just to reinforce this, if we take all of the genes for photosystem 1, it's a 14 subunit complex and 6 light harvesting proteins. Also photosystem 2, again, 20 or so subunits, and then multiple light harvesting proteins. And then we just look at this plot here. Again, if we look at the average downregulation of photosystem 1 versus photosystem 2, you can see very clearly how iron deficiency has a more dramatic impact on photosystem 1. So severe retrenchment of photosystem 1, again, in both Dunaliella salina as well as Tertiolecta. Okay. So for those of you who don't know what photosystem 1 looks like, a cartoon here, this is looking at photosystem 1 from the top of the membrane. So you're looking down at it. And what you see here are the 3 4-iron 4-sulfur centers in red and yellow. In gray are the PSA subunits. So these are the polypeptides of the reaction center of photosystem 1, where the primary photochemistry occurs. And then in black, these are the associated light harvesting subunits, 6 genes coding for 6 polypeptides. You have a dimer here and then a tetramer in the bottom. So we mapped our protein abundances onto the structure just to see what was affected. And this was the result we got. So again, this is just a heat map mapping the polypeptide abundance onto the known three-dimensional structure of photosystem 1 from Dunaliella. And you can see that all of these subunits are about equally depressed. However, what the big surprise was that 1, 8, and 7 shown here. So this is the tetramer. So 1, 8, and 7, so 3 of the 4 subunits actually don't change at all. So we thought, well, that's a bit peculiar because was well established in the literature that when these things are retrenched, everything is retrenched. The entire complex is retrenched, but we didn't see that here. Okay, and then also I told you that there was this protein called TD that was upregulated, and again you see that it's very dramatically. This is a log2 scale; is very dramatically upregulated. So the question is, what's going on here, and what is TD? So when you look at the sequence alignment of TD, you see that it's very similar to a protein called LHCA3. That's one of these antenna proteins that associates with photosystem 1. However, there are very distinct differences. You can see in this, between these two helices, there's this motif here that marks TD, and there's also a larger N-terminal loop. So it's very easy for us to distinguish TD from LHCA3 in genome sequences. In fact, when we looked at genome sequences of other algae that had not previously been known to have TD, the reason was they were annotated as LHCA3 because that's what was known in the literature. But because we knew about the structure of TD, what we found was that a number of algae actually had both flavodoxin, shown in red here, as well as TD. So they kind of co-occur in genomes, suggesting that they function together, presumably in this surviving iron deficiency mechanism. And you can see in this protein similarity network that all the LHCA3s cluster together, and then all the TDs cluster together. So these are clearly, distinct, belong to a distinct protein family compared to LHCA3. And they are reciprocally expressed. When you look either at the transcript or the protein level, you can see that LHCA3 is very abundant in plus iron, shown here, 2872, whereas TD is, on the other hand, very abundant. So this is 5,000 FPKM. So those of you familiar with transcriptomics, this is a hugely abundant transcript in iron deficiency. Okay, so in yellow, this is the actual structure of LHCA3. So we used AlphaFold and we mapped it onto this structure. And you can see that the helices of TD map exactly onto the helices of LHCA3, but there's also these other pink extra bits. So my student, Helen Liu, decided that she wanted to understand what the structure of photosystem 1 looked like from iron deficient Dunaliella species. Now, this was actually, a difficult problem because, of course, I told you that photosystem 1 is retrenched, so it's not as abundant, and of course, you don't get as much biomass. But nevertheless, she went ahead and purified thylakoid membranes from both iron replete versus iron deficient Dunaliella cells and separated all the complexes. And we could already see at this level that there was a difference because the photosystem 1 LHC1 supercomplex, shown by the black arrow, migrates a little faster, meaning it's a bit heavier and perhaps bigger in iron deficiency compared to iron replete. Anyway, and then she purified these complexes. Again, you see the same thing, dodecyl maltoside as a detergent or Triton X100. Okay, so we're suggesting that we have a bigger complex. So with the help of Eva Nogales' lab, so my lab does not have any expertise in cryo-EM, but Helen learned how to do it. And with the help of Eva Nogales' lab and a postdoc who trained her, Helen was able to solve the structure of the iron replete complex from Dunaliella, as well as the iron deficient complex. And again, I'm showing it to here, to you here, looking from the top down. And you see this sort of triangular wedge, which is the photosystem 1 reaction center, the dimer, and then the tetramer here in plus iron. Whereas in minus iron, we see a slightly modified structure. You see the dimer and the tetramer here. However, the big surprise was that we saw a second tetramer associated with the iron deficient complex. And we had a good enough resolution so that we could chain trace and identify what these subunits were. So we had 1, 8, 7, and 3. just like we have in the plus ion structure, 1, 8, 7, and 3. But we have a second tetramer where 1, 8, and 7 are reused, but now we have TD. So TD essentially is nucleating the second tetramer. Okay, so we're making a larger antenna in iron deficiency. And for those of you who remember some of the work done in cyanobacteria more than 2 decades ago, it was shown there that in iron deficient conditions, some cyanobacteria actually make a much larger antenna by using a novel protein that was called CP43 prime. They make a ring around a trimeric photosystem 1 to increase antenna size. So this reminded us of that, and we suggest that this is sort of an example of convergent evolution where these cells, these eukaryotic cells, have solved the problem in a novel way by generating this TD protein derived from LHCA3 that increases the antenna size, presumably to compensate for the fact that they've had to decrease the amount of photosystem 1 they have, and to ensure that they get enough linear electron flow through the photosystems. This is the solution. Okay. So just again, looking at different light harvesting complexes, I just wanted to show you that, you know, there's different ways to skin a cat. So here is the structure from Chlamydomonas. This is not our work, it's from the literature. So again, the photosystem 1 triangular sort of wedge here, the reaction center, the dimer, 1 tetramer, and a second tetramer here. But you'll notice the arrangement is different from that in Dunaliella. And the way that Chlamydomonas does it is actually it has 9 genes for light harvesting antenna proteins instead of 6 that are found in Dunaliella. You have your 1, 8, 7, and 3, and then you have 1, 4, 6, and 5. So they actually have different genes and a different mechanism for putting things together. Here's the Dunaliella one I showed you, and here they're just reusing 1, 8, and 7. And making a new TD here. And then this happens to be one from moss. And again, you see tetramer here and a second tetramer over here. And you'll notice that its arrangement is different too. And in this case, it was by gene duplication of like existing polypeptides that there were. So clearly there's different ways to make larger antenna, and we've identified a new mechanism here. Okay, so from the first part, the take-home messages are, I showed you that in iron deficiency, the most dramatic impact is on ferredoxin and on photosystem 1. This was sort of well known in the literature, and we see it again in Dunaliella. But Dunaliella has these mechanisms for compensation by antenna reorganization, making larger antenna using this novel protein called TD, which co-occurs in algal genomes with flavodoxin. And we estimate that the replacement of ferredoxin by flavodoxin saves the cell 10 to the 8 iron atoms in each cell. And that's why it can survive on lower iron compared to Chlamydomonas. So we'll switch now to some work that originally came out of discoveries in Chlamydomonas and Chromachlorus. And then I'll show you how we undertook some mechanistic studies using a third alga. So this was a discovery of polycistronic messenger RNAs in eukaryotes. And you might remember from what you studied in your textbooks that prokaryotes have polycistronic messenger RNAs and a particular mechanism of translation. But in eukaryotes, one mRNA makes one polypeptide, typically. And we challenged that dogma through our discoveries in algae. So this came about a few years ago when Sean Gallagher was doing some analysis of algal genomes, and he found that some of the proteins that should be present in the genome were missing. So he did some manual curation, and what he discovered was that they didn't show up in analyses because they were found in a polycistronic or actually bicistronic arrangement, as shown here for this particular locus, CRE16, shown in light green and dark green here. The 2 separate proteins, but when you look at the RNA reads from isoseq, this is single molecule sequencing of RNAs, you see in this blue sequence here, a single RNA that covers both open reading frames. Now, of course, it could be that these RNAs are then processed into single monocistronic RNAs for translation. But in this particular case, we never ever saw single monocistronic versions. When we did some ChIP-seq experiments using the H3K4 trimethyl lysine mark, we saw association only with the upstream ORF, suggesting that we have a single promoter driving the expression of this RNA that covers two open reading frames. Okay, so of course, we can also look at the 3' end, looking at the occurrence of the polyadenylation signal. And if we look now at a genome-wide level, I just showed you, one example of that at one locus. But in fact, the promoter mark, we see that, on all of these bicistronic messenger RNAs, and there's hundreds of them in algal genomes, I should say. And then when we looked also at the whole genome level, at these bicistronic messenger RNAs, we found that the poly A signal and also the poly A tail was associated nearly exclusively with the downstream open reading frame. Okay. And when by this point, we had done maybe more than 1,000 experiments for transcriptomics in Chlamydomonas. And if we look pairwise at two adjacent open reading frames in the genome and ask what is their pattern of expression, and we look at the Pearson correlation coefficient, it's 0, because there's really no reason that two genes that are next to each other have to be co-expressed. But when we looked at the two open reading frames on these bicistronic messenger RNAs, they were always co-expressed, which of course makes sense because they're only found on the same transcript. So then we asked the question that, does this messenger RNA actually translate to proteins? Do we get two proteins from it? And we tested that in vitro. This experiment was done with Steve Thegg at UC Davis, because I had just moved and we didn't have a license for radioactivity in my lab anymore. And so we synthesized these RNAs in vitro, and then we sent them to Steve's lab, where he just used an in vitro translation kit. We did it both in reticulocyte lysate as well as in wheat germ extract, so both animals and plants. I'm only showing you the result from the wheat germ extract. And here we're using 3 RNAs from Chlamydomonas and 3 from Chromachloris. Makes no difference. In all cases, we saw both proteins of the expected size from all of these RNAs, suggesting that, in fact, we can translate them in vitro in these heterologous systems. So they're translatable mRNAs. And at this point, we, of course, did note that the upstream ORF tends to be small. You can see they're typically around 8, 9, 10 kilodaltons in size. And then, of course, the last question is, do we find these proteins in vivo? And here we took advantage of large proteomic data sets that we have. And again, I'm just going to show you one locus here. It's I think the same one we looked at before. And what we saw was that the upstream ORF shown in light green, we can identify the most C-terminal peptide in that sequence. And with the downstream ORF shown in darker green, we can identify the most N-terminal peptide in that sequence. And this was important because it told us that the two proteins are actually two separate, different proteins. Because if the sequence in between had been like spliced out and it was like a, one single polypeptide, we wouldn't identify the most C-terminal and N-terminal polypeptides. And again, if we look at the whole genome level, we see this is true of the in general of these bicistronic messenger RNAs. I should say at this point that all of the analysis I'm going to show you, we're going to do only with the bicistronic messenger RNAs that are found exclusively bicistronic, because in fact, some of them are processed to give monocistronic versions. And we did this for both Chlamydomonas as well as Chromachloris. And we saw that the N and C terminal peptides of these bicistronic ORFs were recovered at the same frequency as all monocistronic genes. Okay. And then finally, we did this work in Chlamydomonas and Chromachloris, but we wanted to know whether this was true in other algae. And in fact, we found, as we span other algae, that about 25% to a third of all of these bicistronic open reading frames are also conserved are potentially bicistronic in other algae as well. We don't have isoseq data for all algae, so we can't, say that for sure, but they're next to each other and possibly bicistronic. This is one that's very highly conserved, TOM22 SDHAF3. That's the mitochondrial import component, translocase of the outer mitochondrial membrane. And this is succinate dehydrogenase assembly factor. And we're going to be looking at that later. So at this point, Marco Duenas joined my lab, and his question was, well, you have like this type of bicistronic messenger RNA, how are the two proteins translocated? Because you might remember that the model for translation in eukaryotes is that 40S subunit of the ribosome comes along. It sees the cap structure on the messenger RNA. It then assembles with initiation factors and the initiator methionine tRNA, and it identifies the initiator or the start codon by then scanning for an AUG that's in a favorable context. And that favorable context is determined by the so-called Kozak sequence, which determines the probability of a translation initiation at that AUG. So typically, if this ADS ribosome would assemble here, seeing a favorable AUG, it would translate, and then when it gets to the stop codon, it would dissociate. So how does this guy ever get translated? So of course, we know that viruses have this type of polycistronic structure, and in that case, there are internal ribosome entry sites that allow the ribosome to come in and translate the downstream open reading frames. But we had no evidence for such sequences. In fact, these intervening sequences, their lengths are very different. The shortest one was 1 nucleotide, and then the longer ones can be 200 or plus nucleotides. We didn't have any evidence for like 2A peptides or these types of unusual mechanisms for translating more than one protein. So what I'm going to show you then is that what happens with these bicistronic messenger RNAs is that at a certain frequency, the 40S complex skips ORF1 and goes downstream to ORF2, or leaky ribosome scanning. And to address this question, we switched to another alga, Oxynochlorella protothecoides, which is, as I said, in the tributziophyte lineage. But the reason it was a useful organism for us was because we could do homologous recombination, just like in Saccharomyces cerevisiae. So, for instance, if we have a selectable marker, this was the first one that we used. This alga actually needs a vitamin supplement in the growth medium. This is true of many algae, actually. They need, some of them need vitamin supplements. This one needs thiamine as a supplement because it's missing this gene Thi-C. So we can use that as a selectable marker. When we have flanking DNA, in the case of Oxynochlorella, we typically use 2 kbs, but it can be as small as 500 base pairs. And then if we use flanking DNA from our target gene of interest, we can use homologous recombination to replace the target gene with our Thi-C marker. So that was a big advantage, which meant that we could make mutations in constructs, and we can introduce them into a single site, so that we don't have to worry about position effects as we have to do with Chlamydomonas. The other reason this alga was useful is because it has a small genome. And when Rory Craig was a postdoc in my lab, and he sequenced it, it's a very high quality assembly, telomere to telomere, including centromeric regions. We found it only has 7,500 genes, which is less than half of what's present in, say, Chlamydomonas or Dunaliella, for example. And remember, yeast has about 6,000 genes, so it's the same level. But alga also has a whole chloroplast and all of its constituents. So really, it's very much like yeast. So we can knock out genes, knock them in. We're trying to recycle markers. We can generate allelic series. So it's very useful for this type of functional analysis. And then the other thing that was interesting about it, and this was Jeff Mosley had brought this to our lab, is that it shows exceptional triacylglycerol accumulation. And why this was important is because we could get money for the project by going to the Department of Energy, right? So we can grow the cell either phototrophically, and where it has a typical cup-shaped chloroplast, and you can see these green cells in the microtiter plate, or if we deprive it of nitrogen, and give it a source of carbon like glucose, it will make 60% of its biomass will be oil or triacylglycerol. Okay, so let me show you then what we did with it. Okay, so the first thing we did was derive a Kozak sequence for algae, because this was not in the literature. So what Marco did was he took half the genes in the Chlamydomonas or in the Oxynochlorella genome and asked, what do the sequences around the AUG look like? And it was useful that we had proteomic data, so we knew what the what part of the open reading frames actually were the initiator codon. So for time, I'll just ask you to look at the Chlamydomonas data. So this was the Kozak consensus Kozak sequence that he came up with. This is around the AUG and the mRNA. And then he used the other half of the open reading frames in the genome to test for the prevalence of the Kozak sequence. So the monocistronic open reading frames are shown in pink here. And you can see that they have a certain bit score. So this would be a good Kozak sequence that allows the 40S subunit to recognize. And then in green and blue are the ones from the bicistronic messenger RNA. In green is for ORF1, and then in blue is ORF2. So what you see is that ORF1, so that's the more upstream ORF, has a weaker bit score, and then ORF2 has the stronger bit score, okay? So that suggests that the ribosomes should skip ORF1 more frequently. All right, so how do we test that? So this is then we went exclusively to the Oxynochlorella system here, where we would manipulate the Kozak sequence on ORF1 to see if it influences the expression of ORF2. So we actually used two different bicistronic messenger RNAs, but I'm only going to show you data from one of them. The TOM22 SDHAF locus that I showed you before was conserved in most algae. And then this dot here shows you where we made the mutations in the Kozak sequence of the upstream ORF. And then we replaced the downstream ORF with the reporter gene, in this case of Venus, so that we could quantify expression from the downstream report. So EK means the endogenous Kozak, so whatever nature gave us. And then we could mutate it to better resemble the consensus Kozak, so that would become a strong Kozak. Or we could mutate it to make it even weaker than it already was, so that would be WK for weak Kozak. We could also completely eliminate the start codon, the AUG. So now we've turned it into a monocistronic messenger RNA, right? And then the star shows you that we can actually introduce another methionine into the sequence. And I forgot to tell you that when Marco was doing bioinformatics on all these loci, what he noticed was as I said, the upstream ORFs are small, but what he noticed is that they also don't have any other methionine in the coding sequence. Okay, so there's selective pressure to keep AUGs out of that protein. Furthermore, when we looked at all three reading frames, including the intervening sequence, there are no AUGs in any of the 3 open reading frames. Okay, so anyway, he put one in to look at what impact that would have. So here is the data. We're looking at expression of the Venus reporter now on the y-axis, which actually is broken here. So you see the endogenous Kozak gives you a certain level of expression of ORF2, that's the downstream ORF. If we make ORF1 Kozak stronger, we inhibit expression of the downstream ORF. If we make it weaker, we actually increase expression of the downstream ORF, right? As we would expect according to the leaky ribosome model. If we put in, if we remove the start codon from the upstream ORF entirely, then we get huge expression of the reporter, just like a monocistronic control would give us. And then if we insert this methionine at the star position here in the intervening sequence, then in fact we dampen expression from the downstream Venus, because we've introduced this, unnecessary AUG there. All right, so this kind of suggested that our ideas about the Kozak sequence were correct. The next experiment, what we wanted to do was to actually look specifically at expression of the upstream ORF, which in this experiment we didn't do because we had no antibodies against TOM22. So what the next construct I'm going to show you, Marco replaced TOM22 with another reporter. So in these constructs, and I should say we introduced them all into this neutral locus in oxynochlorella. It's the D-amino acid oxidase, which as far as we know, the organism doesn't use, at least in the lab. And what he did was he replaced the upstream ORF with the GFP reporter, fluorescent protein reporter, and the downstream ORF with luciferase. So both of these can be separately assayed. He made a synthetic green fluorescent protein that doesn't have any methionines in the coding sequence. Okay, and then again, we do the same thing. We look at the endogenous Kozak, weak Kozak, or strong Kozak. Okay, so let's see the results from that. So just focus your attention on the green bars. We had actually used an inducible promoter. So when this promoter is on, we get this green level of expression. So I'll focus your attention on the endogenous Kozak first. This is the GFP luciferase construct. So when we look at GFP fluorescence, we get a certain level of expression from this endogenous Kozak sequence. If we weaken the Kozak sequence, we get less expression. And then let's look at the downstream luciferase now. When we have the endogenous Kozak, which is a good Kozak sequence, you get very low expression of the luciferase. But when we weaken the upstream open reading frame Kozak, we get much higher expression of the luciferase. So we're seeing like this antagonistic relationship between the two open reading frames. All right, so take home 2 is I've shown you that there are dozens of polycistronic messenger RNAs encoded in green algal nuclear genomes. Some of these bicistrons are conserved over hundreds of millions of years of and we think the mechanism for translating 2 open reading frames in eukaryotes is leaky ribosome scanning. And this works because ORF1 is small, doesn't contain other methionines except methionine codons except for the initiator methionine. And I showed you that one can manipulate the ratio of the 2 proteins by making mutations in the Kozak sequence. And we can, in the lab, we can get up to a 6-fold range of protein expression by manipulating these sequences, which may be useful for synthetic biology. And I think this has actually broader implications because now in eukaryotes, many people are paying attention to these small, what they call UORF, so upstream open reading frames. And we think that maybe many of them may be translated in vivo, or they may serve a regulatory role in terms of translation of the downstream. Okay, so I have time to talk about like the third project. The third project was one that we started because many of the students and postdocs who join my lab now say they want to do something useful. And I think that discovery is so useful. So we decided to work to use oxynochlorella for synthetic biology. And the product that we chose to make was human milk fat. So this is something that's put into formula. 50% of the calories that infants consume comes from fat or triacylglycerol. So just to remind you, this is 3 fatty acids esterified onto a glycerol backbone. The fat in human milk is actually a very complex product, and very few people have tried to use synthetic biology to make a complex product like this. So this is just an example. This just shows you fractions from human milk fat, with each of these blue bars in this heat map being actually a different molecule. So what's different about the molecule is that you can have different fatty acids esterified on the glycerol backbone, for example, C10, C12, C14, etc. And this logo is showing you the types of fatty acids that you see in human milk fat, the abundance of different fatty acids, and then the position at which each fatty acid is esterified onto the glycerol backbone. It's C1, C2, or C3. And this particular combination, oleic, palmitic, oleic, is the one that's very abundant in human milk. Okay, so both issues are a problem in formula because the fat typically that comes in formula comes from vegetable oils or palm oils. So what's wrong with it is that instead of OPO, you can have this type of structure instead that comes from vegetable oil. The enzymes in our intestine or in the baby's intestines hydrolyzes the fatty acid at the C1 and C3 position, releasing oleic acid or palmitic acid. And palmitic acid, because it's fully saturated, is less soluble. And furthermore, it binds calcium. So this deprives the infant of calcium as a nutrient. And also the baby gets stomach cramps because the C16 fatty acid is not soluble. So that's one problem. The second problem is that if you look at the fatty acid composition of vegetable oils, you'll see very little of these so-called medium chain fatty acids that are shown in blue in human milk fat. So this would be the C10, C12, C14 species shown here in light blue. So right now, the problem is under the radar in the United States, and we think that's because it's pretty much low-income families who are consuming formula because they don't have, time to take time off work or to express milk and so on. So it's kind of under the radar here, but it's actually something that the Chinese are investing in, and there are companies, two of them, Betapol and InFat, are actual products on the market. And the way they do it is by chemical hydrolysis of vegetable oils, and then they use enzymes to re-esterify the desired fatty acids to give you the desired structure. But of course, this is expensive, so that's one problem. And the second problem, of course, is an environmental problem that, we probably should be using less palm oil rather than more palm oil. So this was one motivation. And the other motivation was the fact that these are data from US, from the CDC, which shows that like by 6 months of age, 75% of US mothers actually using formula. And in fact, the CDC recommends that up to 6 months of age, infants should be exclusively fed on breast milk, if it's possible. But there are mothers who don't produce enough milk either. So the second issue, of course, is feasibility. And if we do this engineering, is this even practical, right? If we do the engineering in algae. And this was something that that I saw during the pandemic, actually, because you're forced to stand in line at the supermarket to buy your groceries. And I was standing in line at the oil and vinegar line, and I saw this product, Thrive Oil. I bought it because it was on sale, half price. And I knew that it was 18-1 fatty acid, so not polyunsaturated, so it wouldn't actually spoil even though it was close to the expiration date. So I bought a bunch of it at half price, and it was a fantastic product. I do highly recommend it. High smoke point, no flavor, so you really taste the vegetables that you stir fry. Anyway, so I thought if you can put it on the supermarket shelf, you can put it into formula, because that's basically the same thing. So John Lin joined my lab, and he had experience in sort of lipid biochemistry, and his goal then was to turn oxynochlorella protothioides fat, shown here in this color scheme, the different fatty acids, into human milk fat. So his first goal was to increase these blue guys, the medium chain fatty acid. So now I'm going to remind you how fatty acids are synthesized. The synthesis occurs using acetyl-CoA as a substrate, 2 carbons at a time. It's attached, it's built onto a protein called acyl carrier protein, 2 carbons at a time. And then an enzyme comes along and cleaves off the growing fatty acid chain from that acyl carrier protein. That enzyme is called the thioesterase, and it has substrate specificity for the length of the chain. So what John did was identify thioesterases, he's abbreviated that as THE here, that have chain length specificity for C12, C14, C16, etc. So the idea was that we could introduce these thioesterases from plants. So the first one he used was from this organism, Cuphea wrightii, it's some kind of weed, I think. And this shows you literally his first experiment. He put the thioesterase into oxynochlorella using the genetic methods that I showed you previously. And you can see that we had very little C14 before. Now the peak is a bit bigger. And C12, which we could never even detect, he was able to see right away. when he added a transit peptide from the alga onto the plant sequence to target the protein to the chloroplast, because that's where fatty acid biosynthesis occurs, you can get bigger peaks and more of the product. And of course, this enzyme is dominant because once you cleave off the chain, that's it, right? You can't elongate any further. So this is the result from that experiment. You can see this is human, this is ape, the original wild type strain, and then these are the two engineered strains. You've got a lot more of the blue product. Okay. He did the same thing with the brassica thioesterase that had substrate specificity for C16 and C18. And you can see that here. Now the goal was to make more of this red one, which is the C16 fatty acid to try to match human. And then when we introduce them both together in one strain, trait stacking, you can see now that we're getting closer and closer to human. So the next strategy was, okay, we've done this with respect to the chain length, and this was all done within a year. What about the second problem, which is to put the palmitic acid on the C2 position instead of the C1 and C3 position? So here's olive oil. You'll notice at the C2 position, it's all 18.1, whereas in human milk, it's mostly 16.0. That was everything. So the way that this was achieved, again, was by humanizing the algal enzyme. So we knew that the human enzyme had substrate specificity for C16. It turned out by chance that the chlamydomonas enzyme also had substrate specificity for C16. These are enzymes in the endoplasmic reticulum. And so what we did is we used the gene replacement technology here, and we replaced the endogenous algal oxynochlorella LPAT. That's the name of the enzyme, lysophosphatidic acid acyltransferase, that puts the fatty acid at the C2 position here. We replace that with with the human or the chlamydomonas enzyme. And again, like in the first experiment, you can see here's the wild type. You have in gray, it shows you the fatty acid at the SN2 position, and we're looking at C16. You see it's very little. When we look at total lipid, including membrane lipid, of course, there's plenty of C16, and that doesn't change in these transgenic lines. But you see now that at the SN2 position in triacylglycerol, you have huge amount of C16 fatty acid. So after using these design, build, test, learn cycles in synthetic biology, this is where we're at right now. Again, you can see that these are the things we were trying to target: human milk fat, starting wild type strain, and now the engineered alga. And you can see we actually have more of the medium chain than we want, but I think we can tune this down. We hit C16 right on the nose there, 19.3%. We're working to try to increase the C18. And then when we look at what fraction of the triacylglycerols have C16 at the SN2 position, and human milk fat, it's about half. And again, we hit that just right. We have about half. And then when we ask what fraction of the C16s are at the SN2 position, because we don't want them at 1 and 3, we see that's the bulk of it in human milk fat. And we also have like nearly 90% of it in the alga. So I think I've sort of only have a few minutes left here, but I just want to end by saying we're going forward with this project. We want to try to customize some of this triacylglycerol composition of alga, for example, by introducing this particular fatty acid because it's required by premature infants who typically do need formula. We can try to make race-specific or population-specific lipids. For example, Filipino populations have more of the OPL instead of the OPO, and we can try to accommodate that as well. And then it's possible to even maybe try to synthesize these specialized fatty acids that have signaling roles in humans by introducing the genes into the alga. Okay, so to conclude this part 3 then, I've shown you that oxynochlorella is really a great system for synthetic biology. We just started working on it like less than 5 years ago, and you can see the advances that have been made. We can make a complex product such as human milk fat by iterative state tracking. So we've been able to humanize the alga to make a product that may be useful in formula. So in conclusion, then I showed you like 3 different projects. The first part was fundamental discovery, relating to photosynthesis. or relating to, general molecular biology mechanisms in eukaryotes. And then finally, how one can apply our basic knowledge to something practical. So I've acknowledged the people who did the work as I went along, but I'll just point out here, Sean, who did the bioinformatics. John was involved in the lipid, the human milk fat project. Marco in the bicistronic messenger RNA translation, and then Helen did the photosystem 1 cryo-EM structure in collaboration with Radhika, who was a postdoc in the Nogales and Niyogi group. And then Jeff Mosley was the one who brought the oxynochlorella system to our lab and supervises much of the oxynochlorella work. And Masha Iwai actually was the one who trained Helen to make all of the thylakoid membranes. So thanks for your attention.
Any questions? Oh. You can go first. Oh, okay. Going back to when we were doing the, I can't even pronounce the word,
Yeah.
How did you realize that was occurring? Like, did you purposely look at the RNA transcript?
That's a really good question. So when one sequences a genome, you use the algorithms for predicting where the open reading frames are and so on. They're actually trained to find only one open reading frame per mRNA, right? So what happened is we didn't find, and then what you do is you run something called a Busco analysis to see how good your genome assembly is, because there are certain genes or proteins that are known to be present in all eukaryotes. And so you want to get like 98, 99, 100% Busco, and we were not getting that, right? And we knew we had a good quality assembly. So you're like, what went wrong? So Sean in my group, he looked at what proteins were missing in our Busco, because we thought, maybe this organism doesn't need these proteins or something, right? But it seemed like TOM22 is an essential protein in mitochondrial import. So how could it be missing that? So he manually looked for that protein and others. And what he found was that they were in this bicistronic arrangement, and that somehow the algorithm had skipped them, right? So he realized that, we need to do something to look for them. So he actually initially manually looked for all of these things. And then we found there were more and more of them, right? So I think that more are going to be found in genomes as people do more of the iso-seq. That's what kind of gave us the clue. When he looked at the iso-seq models, he could see 2 open reading frames on one mRNA. So that's how they were discovered.
Thank you.
Yeah.
And continuing with the bicistronic mRNA, do you think that there might be any role possibly on UTR structure or mRNA structure for that?
Yeah, so we looked, because that was the initial thing, or there must be some secondary structure or something that, but we didn't find anything. I mean, I'm just guessing that this is something just left behind, during evolution, right? Like in the endosymbiont, everything was prokaryotic and polycistronic arrangement. The genes were transferred to the nucleus. They picked up all of the nuclear genome structure. And then these are things that haven't, fully evolved yet, right? And that's why we see sometimes many of the same bicistronic messages in these eukaryotic genomes. So I don't think there's any reason for them to be bicistronic, because they aren't always, as we look across hundreds of millions of years, in many algae, they are not. So they don't have to be. But I think they're left behind. But there has to be a mechanism then to translate them. But it also speaks then to, these upstream ORFs that are found now widely in eukaryotes, that they might be regulatory.
So most of those genes came from originally from the mitochondrial chloroplast.
Well, yeah. So you're wondering whether there's a So that was actually our original thought, was that, yes, maybe the protein products are targeted to the organelles or something, right? But first of all, I guess all of you work on algae know that this is the case. Many of the genes in algae, we have no idea what their functions are, right? Most of them are proteins of unknown function. So we can't really tell if there's something enriched towards the organelle. So the ones where we do know function, it doesn't seem that way. It may appear that they seem to be enriched for proteins involved in nucleic acid transactions, but this might go away once we find out the functions of all of these open reading frames. But right now we can't really tell. Yeah.
Yeah, as far as great talk. So talking about the bicisterone.
Yeah.
Did you look at the GC content or whatever that is it a bacterial, for example, gene transfer somehow?
Yeah, no, so the GC content is not different than any other gene. Yeah. Mhm.
Other question is about your out of frame AUG.
Yeah.
Is ribosome coming and binding and blocking it or something? How does it regulate?
Yeah, you mean when we put one in synthetically and it blocks it, right? No, I think what's happening is that we put that AUG in at a certain frequency, it gets recognized. And of course, it doesn't code for anything, but the ribosome then dissociates and it's done, right? And then because we don't have another internal ribosome entry site, it has to wait for a new ribosome to come along. So it's just really inhibiting translation.
If you put in frame, what will happen?
Same thing, probably.
Same thing.
Same thing, probably, yeah. I think Marco has tested that, but I don't remember the data off the top of my head. I would expect the same thing, yeah.
One last question, we should wrap up.
Thank you. So beautiful talk. I was curious about the lipid and changing the fatty acid content so substantially. How does the organism do?
Yeah, that's a really good question. I should have mentioned that. So when we do the growth curves, they're a little bit slower, but they catch up. So there's definitely an effect. And that's probably because some of these fatty acids are getting into membranes as well, where they don't belong. And so it's affecting membrane function, such as, bioenergetics and so on. And actually, this is the reason why it's actually better to do it in a unicellular, simple eukaryote rather than to do it in plants. So Peter Eastman's lab has actually, driven much of our thinking on this project, because he has tried it, I think, in Camelina, maybe even Arabidopsis. But there are developmental problems, right? Because you have to, you engineer the organism, but it has to grow into a whole plant and go all the way to seed, right? Because that's where the oils are extracted from. They're extracted from seeds. So it really is not practical to do there because the plants don't develop properly. And a lot of fatty acids are also involved in signaling and so on, right? So in that sense, it's better in this system. And these types of approaches have also been used. I think I saw a little press release from a company recently where, but I think our table looks better than what they put in their press release. They did it in Yarovia lipolytica, which is a yeast that people use for engineering fats. So I'm guessing that they use some of the similar approaches, although they didn't say how they did it. But yeah, so they also went with a yeast. But we're thinking that this might be, in the long term, cheaper because we could, with the yeast, you have to use sugar. And we've done this heterotrophically here as well, just to go faster. But in the long term, we could grow these things photosynthetically, right? So you can use energy from sunlight, which is better than using sugar that comes from sugarcane, because then we're back to the environmental problem, right?
Thank you all for coming. And if you have any last questions, please come up afterwards.